Some say that they're a miracle of modern medicine. Some say they're the focus for a dangerous mass experiment on the population. I'm talking, of course, about the mRNA vaccines. How are they different? What are the critiques? And what's the intriguing dispute at the heart of their origin story? Well, let's have a look. The breakthrough technology behind some of the COVID-19 vaccines is synthetic mRNA. And it may seem like it emerged as suddenly and dramatically as did COVID-19 itself, but like most overnight success stories, it was actually decades in the making. So how does it work? Well, first of all, you have to understand how viruses work. Now, I know many of us have learned about all of this in excruciating detail over the last year, but as a quick recap, viruses attach themselves to your cells using the familiar spikes that you'll have seen, and they inject a piece of DNA or RNA into it, coded genetic information basically. The cells take DNA and follow instructions coded within it, and in this case the virus DNA or RNA gets them to make new copies of the virus. They turn your cells basically into virus factories, churning out new copies to go and find more of your cells to infect. Of course, after a while, a few days generally, the body's immune system begins to recognise the virus as an intruder and begins to produce antibodies. The antibodies connect the virus spikes, preventing them from infecting new cells and marking them for destruction. By then, it has a lot of virus to catch up with, which has been busy infecting you and giving you whatever symptoms are associated with that virus. So the purpose of any vaccine, whatever technology it uses, is to train your immune system to recognise the features of a virus, specifically its type of spike protein. So if it does turn up, the antibodies don't take days to work it out. They can attack straight away and prevent it from getting a foothold. Traditional vaccines do this by injecting a weakened or inactivated version of the actual virus. Such vaccines require samples of live virus to work with, and they can be slow to develop. The mRNA vaccines are substantially different. Rather than introducing a deactivated virus, they aim to get your own cells actually manufacturing the spike proteins associated with the virus. So they produce artificial RNA, the coded instructions to your cells on what proteins to manufacture, and that RNA is specifically focused only on producing the spike proteins. And the type of RNA it uses is a thing called mRNA, which stands for messenger RNA. mRNA was something that we knew should exist before we actually discovered it. Its existence was predicted in 1961 by two French scientists, Jacques Monod and François Jacob. How did they know? Standard DNA lives tightly wound up in the nucleus of the cell, but proteins are made elsewhere in a different cellular compartment called the cytoplasm. So they argued there had to be some intermediary molecule that somehow went from one to the other. So the mRNA of the vaccine carries the instructions on how to build the spike of the coronavirus. It's injected, contained within what are called lipid nanoparticles, which allow for controlled release of the mRNA and minimise adverse reactions. They enter your cells, the cells produce the spike proteins, which train the immune system to recognise it, regardless of whether it's attached to a virus or not. After a few days, the spikes have all been destroyed, along with the mRNA, leaving just the B cells or memory cells, which are on hand to produce the right antibodies should the real virus come knocking. It's fair to say that the arrival of the mRNA vaccines was greeted with a significant amount of hype. How mRNA technology could change the world, and the first COVID-19 vaccines have changed biotech forever. And it does seem to be full of promise. The Atlantic reported that a team at Yale have patented an RNA-based vaccination against malaria. Pfizer has said it will use mRNA against seasonal flu, enabling it to be much quicker on top of the seasonal mutations. BioNTech, the company partnered with Pfizer on the COVID-19 vaccine, is working on therapies that could potentially fight off advanced cancer. It could also be effective against multiple sclerosis. If it delivers, the companies involved could save many millions of lives and do so at an extremely healthy profit. So there's a lot riding on this. Now, it didn't hurt the hype that there's a classic human story attached to the technology, the triumph of a determined individual over seemingly hopeless odds. Let's quickly go through the history, the official version to start with. It's written up in numerous places with varying amounts of colour and human interest, but let's start with the current Wikipedia version. You'll find out why in a minute. The idea that mRNA could be used for therapeutic purposes was first realised in 1989 by researchers at Vical, a Californian biotechnology startup. Researchers working at the Salk Institute that year published an article showing that nanoparticles could transfect mRNA into cells. In 1990, John Wolfe at the University of Wisconsin collaborated with researchers at Vical and reported positive results where naked or unprotected mRNA was injected into the muscle of mice. These studies were the first evidence that in vitro transcribed mRNA could deliver the genetic information to produce proteins within living cell tissue. So that was the initial work to identify a proof of concept. It led to suggestions that the process could be used for immunisation purposes. Lodge that part in your memory. We'll come back to it later. At this point, Hungarian biochemist Katalin Karikó enters the story. She was trying to get the process to work, but found that injected mRNA would be identified as an alien intruder by the immune system and would therefore fail to reach the target cells. She worked on it for years, tried and failed to get grants to support the work. Because it was seen as too far-fetched, people couldn't see the value of it, often even her own colleagues. She said, "Every night I was working, grant, 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 and it came back always, no, no, no." She was consequently demoted because she wasn't bringing in the expected levels of grant money. Now, many people would have given up long before. You can see the appeal of this story, of course, because we already know the punchline. Working with another immunologist, Drew Weissman, she finally found that they could use a modification to the RNA that would successfully get it into the cells without triggering the immune system's defences. They published a paper on this process in 2005, and that work was then picked up a few years later by a stem cell biologist who recognised its importance and ended up co-founding a company to take it forward. That company was Moderna. The name was constructed from the words modified and RNA. A separate group founded BioNTech, the company that went on to take Karikó on board to lead their RNA work. And they raised money. They were seen as being the pioneers of the future. And they researched and they developed lots of potential, but slow progress. Until suddenly, of course, they were poised at the right place when COVID-19 came along. They were able to have their vaccine essentially developed within 2 days of the virus having been sequenced. And that was a compelling success story, one that the media understandably loved to tell. And there was one particular aspect to all of this that a few people did comment on, but which got very much buried under the urgency and the drive to defeat the virus. And that was the fact that here was a new process. I mean, decades in the making, but new nonetheless. Now, it had gone through rigorous blind safety trials, absolutely. High pressure, high profile, nowhere to hide on any of that. But it was still nevertheless a very new process, one that operated at the cellular level and one that was being pushed very swiftly to huge scale. As you might expect with a new technology, whatever the legitimate questions and critiques, and we'll come to those in a moment, some of the early stories circulating social media were alarmist and not particularly well-founded. So, for instance, some people were spreading messages that mRNA vaccines in some way reprogrammed your DNA and turned you into a genetically modified human being, which of course isn't remotely how it works. We should be clear from the description I've already given. It does not have the power to affect the DNA inside your cell. But of course, lots of people didn't know that, and it all sounded very credible to those people. So between the hype on the one hand and the horror stories on the other, it's no surprise that people ended up confused about what to think about any of this stuff. When things become important, people fight over it. The drug companies, including Moderna and BioNTech, have huge stakes in all of this. The new companies have an interest in fostering the hype that they're reinventing the field of medicine, which doesn't mean that they're not, but you probably don't want to just take their word for it. Those whose assets lie mainly in the older technology, they kind of have an interest in encouraging any concerns or doubts about the new, which again doesn't mean there aren't valid concerns. And then individuals also have an interest. Moderna founder Derek Rossi argued that Carrico and Weissman deserve the Nobel Prize in Chemistry for their breakthrough work on all of this. Nobel Prizes are a big deal. That sort of talk also brings out the competitive instincts in lots of people. And maybe that's why Robert Malone entered centre stage. In that conventional history I shared from Wikipedia, you'll remember it began. Researchers working with the Salk Institute published an article showing that nanoparticles could transfect mRNA into cells. How much significance gets placed on that early ground-laying work compared to the work that came afterwards? And amongst that anonymous group of researchers, who was really leading the field and coming up with the innovation? According to Robert Malone, who describes himself as the inventor of the mRNA vaccines on his website, the answer is clear. He tells the story of himself as the driving force for that research. He generously says that he was enabled by being surrounded by brilliant people, but ultimately his story is one of his own genius being betrayed. He led all the research but didn't get the principal credit he deserved because of the toxic politics and the rampant egos. He was on some early seminal patents, but there were numerous others that the institution chose to withdraw and for which he therefore didn't get the credit. In his version, he did the heavy lifting for this new technology. Before people go handing out Nobel Prizes, he would like people to remember that. And all these articles praising Carrico are just sickening to see, he says, because her contribution was really very, very minor. He's told the story in writing and on video. His wife wrote a more detailed version. It comes across as credible in a number of ways. Firstly, he clearly was directly and deeply involved. Yes, the early papers include his name. This seminal paper, for instance, describing the essence of the process. The early patents include his name. Indeed, the earliest paper by Carrico credits his influence. As far as I'm aware, she hasn't responded publicly to his more recent disparagement of her. She's been rather busy by all accounts. Others have been unimpressed by his claims, however, suggesting that his contribution came more than 2 decades ago, was just one part of the technology, and he'd contributed nothing since. And indeed, if you review Carrico's social media profile, you can see someone over recent years fully engaged in talking about endlessly the development of this technology. For him, not at all, until this all burst into recent prominence. Throughout history, we've seen that to be credited as the inventor of a thing, it's not enough to have had the original idea. It has to come with the work and the persistence, the obsession even, to turn it into the useful thing. Nevertheless, his role was clearly not minor. He talks about the history in detail, extremely congruently. Interestingly, though, this isn't just a pitch by a person saying that he deserves some of the credit, or indeed all of the credit, because while establishing his authority in the field, he is using that to attack the outcome. These mRNA vaccines have not been sufficiently tested, he suggests. The spike protein they create is cytotoxic, and that makes them dangerous. And as you can imagine, this has proven to be an incredibly compelling pitch. The inventor of the mRNA vaccines says they're not to be trusted. Well, many people see that and assume that he was de facto the inventor of these vaccines, which of course is not the case. But even so, a critic from a point of deep knowledge. His arguments have been amplified by Brett Weinstein on his channel and blog. He's been interviewed in various other places, all over Twitter like a rash. We'll talk about his authority and his pitch in a moment, but let's look for a moment to the substance of his main critique. The vaccines create spike proteins, as we've discussed. The principle is that it stays relatively fixed in position, If small amounts of the spike protein do come loose, travel around the body, it's quickly carried to the kidneys where it is destroyed. Malone suggests that that is not the case in practice. He argues that the spike proteins are cytotoxic in their own right, which means that they damage human cells, and they travel freely around the body, potentially accumulating in places where you don't want them, such as the ovaries. Now, as I always say, when the details of scientific discussions get complex, I'm not going to pretend to be in a position to evaluate them, make a judgment about what is scientifically correct or not. However, one rule of thumb I have found useful is to remember that the opinions of individual scientists, however well credentialed, is not a substitute for actual research. Now, so far, there's no research evidence available suggesting that spike proteins created in our bodies from the COVID-19 vaccines are toxic. The vaccines are relatively new. You can argue for the possibility of long-term side effects, and you can make the argument, as a number of intelligent people do, that given the novelty of the tech, it really was an act of faith on the part of the government to fund this to scale in the way that it was then going to be used by a large slice of the population. But that's not the same as saying here and now the spike protein is definitely toxic and amplifying that message far and wide. A number of scientists have been quick to specifically state that this is false. Professor Anna Durbin, Professor of International Health at Johns Hopkins Bloomberg School of Public Health, told Reuters that the spike protein created by the vaccines is not identical to that of the virus. It has been modified to be harmless. When you are vaccinated, your cells use the mRNA or DNA from adenovirus vector to make spike protein in your cells. Your cells show your immune system the spike protein and pieces of the spike protein. The spike protein does not kill those cells. It is not cytotoxic. The trouble with speaking so authoritatively is that it is a new process. It's not fully understood. A recent paper illustrated the point because it supported at least one aspect of Malone's argument. They studied the blood plasma of vaccinated volunteers. On the one hand, the study showed that the vaccine had indeed done its job in provoking the immune system response. Viral proteins present were quickly cleaned up by antibodies. But on the other hand, it showed that more of the spike proteins were actually entering the bloodstream than had been expected via a mechanism unknown. It said this: "We hypothesized that the cellular immune responses triggered by T-cell activation, which would occur days after the vaccination, lead to direct killing of cells presenting spike protein and an additional release of spike into the bloodstream. The mechanisms underlying release of free S1 and the subsequent detection of the intact spike protein remain unclear and require further studies." One example of where Malone makes his claim about spike protein and gives a reference is in this tweet. "The SARS-CoV-2 spike protein is cytotoxic. That is a fact. Who says so? Multiple peer-reviewed references. The Salk Institute. It is the responsibility of the vaccine developers to demonstrate that their expressed version is not toxic." The article that he links to is this one, and it starts like this: "Scientists have known for a while that SARS-CoV-2's distinctive spike proteins help the virus infect its host by latching onto healthy cells. Now, a major new study shows that the virus spike proteins, which behave very differently than those safely encoded by vaccines, also play a key role in the disease itself." So the authors of the article believe that the vaccine spike proteins are safely encoded. Therefore, Malone isn't pointing to a study that proves that they aren't. He's just demanding further proof. Which is fine if you're not already insisting with certainty that it's toxic and it causes harm. And it's this maddening mix that we often see, where someone looks like they may have a solid point, but they obscure it with overclaiming or with poor evidence. One thing to note about Robert Malone: there has been a narrative in recent days that there's a concerted attempt to wipe him from history. For example, this video pointing out that on the 14th of June, the Wikipedia page about RNA vaccines we've already looked at had his name prominently in that history section of how the vaccines were created. But just 2 days later, gasp, it had disappeared. He'd been written out of history, probably by the evil corporations who didn't want the vaccine inventors spoiling their pitch. And his conclusion has been all over Twitter since this video went live. But is it true? Lucky for us, Wikipedia has a full edit log of changes that are made to its pages. You don't have to rely on the Wayback Machine. And if you check, you'll find that the section with Malone's name was added for the first time on the 9th of June. And it was then changed back on the 14th by an editor on the basis that it was a clear conflict of interest edit. If you looked at the editing history of the account that made the edit, they had done only one other action on Wikipedia, and that was to create a page for Averick Pharmaceutical. Averick Pharmaceutical is the name of a company that was founded and led by Robert Malone. That page was tagged for speedy deletion because it appeared to be advertising, and the account was blocked indefinitely after the second edit for advertising and promotion. So in other words, Malone, or someone acting for him, edited the Wikipedia page to big up his contribution. The rewriting of history was Wikipedia rejecting the edit. So look, that doesn't mean that Robert Malone doesn't have a legitimate grievance for how history went. Maybe the version that was written into that Wikipedia entry is exactly right and should be the official version. But he does not seem to me to be a reliable narrator. Maybe unfair, but that's what happens when you promote yourself so actively as a victim in quite such a transparent way. Maybe none of it matters. He could be proud and understandably bitter and yet still be right. But some of the things he's been amplifying seem to have low credence, and that raises question marks. So for instance, this tweet just a couple of days ago pushing a peer-reviewed paper that comes to the startling conclusion, simply put, as we prevent 3 deaths by vaccinating, we incur 2 deaths. The paper is this one by Harold Wallach et al. Maybe it's correct, but a cursory review comes up with a few possible reasons to be careful. One, it's published in an obscure journal, which is often an indicator that it couldn't get space in one of the mainstream ones. Suggestive, but it's not on its own conclusive. Second, the principal author is not a virologist, he's a clinical psychologist, and his work has included papers on homeopathy and spiritual healing. Now, you don't have to be a virologist to have insights in this area. Physicists, statisticians, evolutionary biologists, all sorts of people can bring insights to this question, drawing from their own area of expertise. I don't quite get the crossover in this case. And thirdly, the point at the heart of the paper seems to be what has been criticised elsewhere as a sloppy approach to handling the data from the adverse reactions database. A number of stories have been spreading, doing calculations of relative harms based on the number of cases recorded in systems designed to locate instances of severe side effects from vaccination. The stories point to an increase in reports of deaths since COVID vaccinations began. They subtract the total from the former baseline of reports and then put that number forward as the number of people that are dying from the vaccine. The problem with that is that there are a number of people that die anyway in any given week. If you have a program of mass vaccination, that means it'll be natural to see some people who die within a week or two of vaccination, who would have died anyway. Why have the numbers of reports surged in these databases since the COVID-19 vaccination started? Well, you'd expect them to, because there are many, many, many more people in any given country that just received a vaccination. The question isn't whether there's a surge of reports in the database. We expect that. The question is whether the percentage of incidences of deaths is different to the background range of normal deaths. And that seems not to be the case. So you have to wonder at the integrity of someone who is painting themselves as the whistleblower from the point of knowledge, who is prepared to amplify those sorts of figures. Such actions are compatible with someone who's bitter enough with history to act as a saboteur. I mean, a saboteur of the companies that own the patents that he feels that he should be benefiting from, enriching themselves on the back of his work. They don't seem to be the actions of someone utilising their advanced expertise to help solve a public problem. That's unfortunate, because it's not unreasonable to say we should be asking hard questions of this new technology. The crude figures being spread by certain contrarians may not hold up to scrutiny, but we have seen the announcement that the mRNA vaccines have been linked to a small number of cases of heart inflammation, called myocarditis, among young people. So far, the CDC has identified 226 cases among people aged 12 to 24, which is higher than you'd expect in that age group. In a normal time, there'd be fewer than 100 cases. It's obviously a tiny number when seen against 130 million people who have been vaccinated with mRNA vaccines. It's no surprise that such a tiny effect wouldn't have shown up in the trials, although it suggests only a 1 in 500,000 of having this effect, which is in most people's definition of acceptable risk. Of course, you have to understand what the cause is, and to be able to evaluate any other implications, because maybe it's an example that if spike protein does get into the bloodstream, then in a tiny minority of cases, that is what is responsible for causing this harm. Or alternatively, it could be wholly unrelated. There is still no proof that there is causation here. So where does this leave us? 1, the mRNA technology generally seems to have great promise for the future. Huge credit to those that have developed it. 2, is it ideal that it was pushed out immediately to scale in this way? No, it wasn't. But the world doesn't wait until you're ready. The question becomes what you choose to do about it and what level of risk you're prepared to take. What I see all over this discussion is people amplifying risks. One group says COVID-19 is the deadliest thing ever, but will wave aside any suggestion of risk with the vaccines. Another will say COVID-19 is a trivial thing, but they will jump up and down about how risky the vaccines are. These are not serious deliberations by serious people. These are tribal affiliations expressing themselves by proxy. For some perspective, George Washington won the War of Independence against the British partly because he'd ordered all of his troops that hadn't yet had smallpox to be inoculated against it. Now that was an incredibly crude and dangerous program then. You could well die from that. And they just sucked it up, got on with it, and won the war. Now look, most people have already taken their decisions. Some I know, however, still haven't. You should never take medical advice from YouTubers. I repeat it again, I've said it numerous times. But if you're concerned about the newness of the technology behind the mRNA vaccines, i.e. you're on the risk-averse end of the spectrum, it's not an unreasonable concern. And there are alternatives that don't use it. The Novavax vaccine, coming up soon in the US, has had good efficacy, doesn't use the mRNA technology. The AstraZeneca's jabs, particularly in the UK, have good enough efficacy and are likewise based on older technology. Ultimately, the argument for having them is more to do with public duty, preventing yourself from becoming a vector of transmission to somebody else. The risk profile of dying from the disease yourself, if you're healthy, is pretty small. The risk profile of serious problems from any of the vaccines is even smaller. George Washington would laugh in our faces, I dare say, and to some extent I'm with him. If you're looking for certainty, the only consistent message from all of this is that those who offer the most certainty in what they say tend to be the least reliable guides. Look, whatever you chose, or whatever you decide to choose, it's probably going to be fine. Thanks for watching this video. If you liked it, please share with anyone else you think would also enjoy it. Word of mouth is really important to us. And if you've not subscribed yet, what are you waiting for? As the saying goes, that subscribe button won't smash itself.